Hi everyone. I'm Mohammed Farahat from Computational Biology Division, University of Cape Town, and today I'm going to present the African Pangenome Reference Graph project. So the main project aim is to build an African pangenome depending on the non-reference sequence data we identified from short-read sequence variant calling, and also to build customized workflow to identify the non-reference sequences, and to build an African pangenome reference graph, and to build a near telomere to telomere reference genome. So based on the shortcomings of the current human reference genome, the GRCH38, the most common for human variant calling, and also with the knowledge that the African population contains the greatest genetic variation, we aim to build an African-specific pangenome sequence based on the long-read whole genome sequence data to improve the calling of variants from African samples. So we aim to generate high-quality backbio hi-fi data from between 25 to 30 samples representing the major and African ancestral components we have identified based on the short-read sequence data. So we intend to use the latest tool to generate high-quality de novo pseudo-phased assemblies for each sample, and using this sample together to build an African specific reference genome graph. Then we build another African graphs using the HPRC samples and also the GSVC samples. So then we compare between different graphs to assess our graph quality in identifying African-specific variants. So we have 27 African individuals, and we are using backbio hi-fi sequence data. The first batch was 30x coverage, and now we just received 2 weeks or 3 weeks ago the final batch, which is 60 to 70x coverage. At phase one of the computational work in that project is tool assessment and workflow development, is to assess of the tools for de novo assemblies and graph building, and also to build and develop workflows for reproducibility, and to assess of the computational and the infrastructure requirements that required for building the graph and do the downstream analysis. So the second phase is to build the graph and variant calling. We start with generating pseudo-phased assemblies from long-read hi-fi sequence data, and also to assess the quality of the assemblies. Then build the graph. We use two main approaches, the mini-graph cactus and BGGB. And also we build other African graphs from the HPRC sample, African samples, and the HGSVC's African graph. And then we are going to assess the graph quality and explore variation in complex and disease-related regions, and to assess the impact of variant calling against the different graph combinations, and also the linear reference one. So we had a series of hackathons and workshops to be familiar with this technology and the tool, and also set up the tools and required infrastructure. We started, first hackathon was in 2020. It was just basic training for Git and GitHub and workflows and Docker and containers, followed by second hackathon. It was in 2023. It was a face-to-face hackathon here in Cape Town with HPRC people and BGGB team. We had at the time 6 hi-fi samples. Was ready for to do the initial analysis, and it also was opportunity for us to be familiar with the tools and the required infrastructure, and how to set up the tool and how to continue the downstream analysis. We followed that hackathon with many sprints to dive in depth with the downstream analysis. So for the methodology, we use the hi-fiism since we have the hi-fi data. So for hi-fiism, it has 3 main approaches. Hi-fi only assembly, like in our case, or the thereo-binding assembly in case if you have the whole family, so you can phase the paternal and maternal haplotype as well, or the Hi-C integrated assembly. And the recent version just released, I think, 2 months ago for the hi-fiism tool, it's now allow you to combine between the different technology of the long read, so you can combine the ONT ultra long reads, for example, with hi-fi. So we started the workflow, the manual workflow, using bash script at the beginning from the raw read to do the quality check on the raw read, then to build the assemblies, to map back these assemblies, and then to partition and sequence naming to give each haplotype so you know which from which. Before building the Pangenome, also you can partition per chromosome, then you build your Pangenome using BGGB or minigraph cactus. But we decided to move to automated workflow, so we choose the WDL. WDL is workflow description language, and here is a recipe for workflow submission. You need to prepare your tools. You can containerize it using Docker containers. You need to write your workflow itself. It contains your scripts. You can also add bash scripts or whatever the programming language you can include in the WDL syntax, and then you need execution engine like Cromwell or MiniWDL, and then the data itself. And here, how is the WDL is running? You need to submit your WDL, and the workflow itself contains command line bash scripts and also the list of the tools that you are using. Then it uses the execution engine like Cromwell to execute that WDL, so it generates individual scripts and dispatch it on your back end, like if you are using your local HPC or you are using cloud like Terra, for example. And the final version of the workflow. It's full automated, and you just need one single input, which is the, okay, you can't see my pointer, but anyway, that the red arrow, it's just one single input is the raw, the pass to the raw directory, and it will run in parallel the raw data quality check and the assemblies, and after the assembly quality check. And also we notice, since I'm using here, in each single run, you need to pull the latest version of each tool using Docker, for example. So it's important to track the version of the tool. That's why we add the tracker at the end. So in each single run, you have a record of the versions of the tools that you use. And here is how to continue after you're happy with the quality of your assemblies. Then here is how to build the Pangenome itself. So this workflow is to using the BGGB. So first of all, you need to prepare your assembly, and then you need to use one or both of the linear reference. Why I'm going to use the linear reference with the Pangenome? Because it's simply, you can add it on the top like a backbone or a coordinating system to your assemblies. And then you can continue giving the names of the haplotypes and then build your graph. And here also the workflow is if you are interested in a specific region in the Pangenome, so you can subtract this region and visualize it using 1D and 2D and 3D. With the different tools we are using in the graph visualization. And also by the efforts from both team, the BGGB team and Minigraph Cactus, they offer last year, I think it was in November or December, workflows for both of the tools, so the BGGB and Minigraph Cactus. So here is the final workflow, is our workflow to do the quality check of the raw reads, build the assemblies, do quality check for the assemblies, and then we customize the next flows for both BGGB and Minigraph Cactus to be set up and running in our HPC. And then we are using another developed workflow for the downstream analysis and graph visualization. And here was some of the results about the quality of the assemblies for the 30X. At that time, but we are using now the 60X. Since we just received it a few weeks ago, but I can tell with the 60X we find the context length is more than the 30X, and the number of contexts is less than the 30X, which is good. And here was a comparison between our assemblies and the HPRC, the African HPR assembly. And here is to show the single gene and multi-gene retention. As you see in both haplotype 1 and haplotype 2 for our data sets, and also the HPRC data sets, is still in the same level with a minimal difference, which means that both our samples and the HPRC sample can capture successfully the single genes. And for the multi-genes, as you see, it's lower than in both the HPRC and our samples as well, which means maybe it's due to complex and maybe it's complex region. But still, there is no significant difference between our assemblies and the HPRC assemblies. Then last October 2024, we had a workshop. It's Bring Your Own Data workshop. So we invited all the African researchers from different African countries and institutions to come to Cape Town and to get their own whole exome or whole genome sequence data. Then we trained them how to be familiar with doing the variant calling against the graph. And here it was the workflow. So they came with whole genome or whole exome. Then they can do the variant calling against the linear reference and the graph reference, and then compare both the VCF files from the linear and the graph. Here, one of the samples from one of the patients from a neurogenomics lab, a neuroscience institute in UCT. And as you see, there is a significant difference, and almost the majority of the differences between the CHM13 graph and the linear reference graph. As you see, there is a significant difference for the graph one. And as you know, in the reference genome, there are regions which called dark regions. So it can be dark by depth or dark by mapping quality. So also it's one of the patients from Neuroscience Institute. As you see here, it's visualization on IGV for the HLA-DRB5 gene, which is dark by depth. As you see here for the linear one, however, the mapping quality for both of them is 60, but as you see, the coverage here is very poor, which enhances it when we use the graph. And also for the HSPA 1A and B genes, which was darkened by mapping quality. As you see in the linear side, the mapping quality was 0, and in the graph side, it's 1. And also for SMN1 and SMN2 gene, which was darkened by mapping quality, it was 0 in the linear and 1 in the graph. Also, one interesting thing that we just found last month, when we compared the ColorsDB. So ColorsDB is a consortium of long-read sequencing. It's aggregated data from about 1,400 long-read genomes from a diverse group of institutions with ongoing hi-fi sequencing research projects, and also they included other controls cohorts like HPRC and AGSVC. As you see, when we did the intersection analysis, we found more than 33K of, I'm going to finish before a minute. More than 33,000 variant, which is unique for our only 27 African control samples. That means that we really need to address the African diversity, and also it proves the underrepresentation of the African genomic diversity in the public and global databases. Also has a collaboration with the Human Pan Genome Reference Consortium. So we collaborate with them in training and also to access the new methodologies and new workflows. And also we are now thinking how to use our African Pan genome to augment the global reference genome. And also for one of the phases, or the last phase of that project, is to build a telomere genome. So we have 7 samples from DRC with enough blood to perform 3 different type of ONT long-read sequencing. So what is next? What we are doing now is we start to build the Pangenome using the 6x coverage samples. And as I mentioned, the T2T for the DRC samples. And also we are working on to assess our Pangenome and extract the high quality regions and to build different African Pangenomes from the global HBRC Pangenome and also the AGSBC, and to investigate how to contribute with the global human Pangenome and augment it with our high quality regions. And also we can think about the concept of Pangene if you have like enough or acceptable quality for particular genes, so we can subtract it and provide it as a Pangene. Yeah, thank you.
Thanks very much. We have time for one question. If there is a burning question.
Thank you. Yeah, I just want to warn everyone about using only the MapQ statistic, mapping quality statistics, when you're dealing with the long reads. It happened to me that in some datasets I was working on, I had a very long read that was actually genuinely mapping correctly to the reference, only that there was a long insert inside the long read, and actually it was very relevant for my research. And I got a MapQ quality of 0. Though on the 2 ends of the long read, you have very large regions mapping very perfectly onto the reference genome. So in that case, it's important to go look for other indicators in the additional fields in your SAM file that will give you, for instance, the number of matches or the number of inserts, etc., and to work a bit more than just looking at the MapQ file.
Thank you, Bakaya. That's what exactly we are doing, especially for the patient samples. So we were not relying only on the mapping quality. We take this particular region and then go to the BAM files itself, see where is the region, if it's insertion or longer insertion or deletion and so on. So we have further analysis to confirm that. Thank you.